Hello and welcome. Today we are jumping into a field that honestly sounds like it's straight out of a sci-fi movie, but it is very, very real, and it's happening in labs as we speak. We're talking about tissue engineering, basically building biological parts from the ground up. So let's just start with a huge, huge question. What if we didn't have to wait for organ donors? What if we could just build a new heart, or a liver, or even new skin whenever we needed it? It's an incredible thought, right? And it's pretty much the ultimate goal that's driving this entire field of science. So, okay, how in the world do you even get started on a goal that big? Well, the answer lies in this field called tissue engineering. At its core, it's all about creating artificial but fully functional tissues in a lab. And the goal is simple, but it's also incredibly profound: to repair, or maybe even completely replace, parts of our bodies that have been damaged by injury or disease. To really get how we can build something as insanely complex as living tissue, we need to start thinking like engineers. And every engineer needs a construction kit, right? They need their raw materials. So let's dive in and look at the body's fundamental building blocks. You know, any big structure, it always starts with its most basic unit. For a house, that's the brick. Well, for the human body, it's the cell. Every single thing your body does, every movement, every thought, it all comes down to these tiny, microscopic powerhouses. For our project, these are our biological bricks. But here's the thing, not all of these biological bricks are created equal. On one hand, you've got what we call differentiated, or adult cells. Think of these as the highly specialized workers on our construction site. The muscle cells that contract, the nerve cells that fire signals. They're experts, absolute pros at their one specific job, but they don't really divide and make more of themselves. And then you have the stem cells. These are the versatile reserves. They're undifferentiated, which basically means they haven't decided what they want to be when they grow up. Their superpower is that they can divide like crazy and can become almost any type of specialized cell the body needs. Of course, just having a pile of bricks doesn't give you a building, does it? You need something to hold them all together, to give the whole thing structure and strength. In our bodies, that critical job belongs to the extracellular matrix, or the ECM for short. It's literally the biological mortar that surrounds our cellular bricks. And this isn't just simple goo. This mortar is a really sophisticated mixture. It's packed with proteins and molecules that all have specific jobs. You've got collagen, which provides incredible strength. It's kind of like the steel rebar in our tissues. Then there's elastin, which, as the name suggests, gives everything flexibility, so tissues can stretch and snap back. Then you have these glue molecules, like fibronectin and laminin, that help the cells stick to everything. And finally, you've got things like hyaluronic acid to keep it all hydrated. And the biology here is just so clever and specific. For example, buried inside that fibronectin glue is this tiny little sequence of amino acids called RGD. The best way to think about it is like molecular Velcro. It's a specific chemical shape that cells have receptors for, so they can literally grab on and create this really strong, secure bond with their surroundings. For engineers trying to build tissue, getting these tiny details right is absolutely everything. Okay, so we've got our bricks, the cells, and we've got our mortar, the ECM. Now, where does all this construction actually happen? Well, it happens outside the body in a super controlled environment. Welcome to the workshop. The foundational technique that makes any of this possible is something called cell culture. It's basically the science of keeping cells alive and, happy outside of the body, or in vitro, as scientists call it. I'm not exaggerating when I say that without the ability to do this, the entire field of tissue engineering just wouldn't exist. And, creating a happy home for cells is not easy. You essentially have to trick them into thinking they're still inside the human body. They need a surface to stick to. They need a liquid nutrient bath, which we call media, that's full of sugars and salts to food on. And the atmosphere has to be perfect, exactly 37 degrees Celsius, with a very specific mix of CO2 and oxygen, and super high humidity so they don't dry out. It's a delicate balance. All right, we have our materials, we have our workshop. Now we need a plan. We need a blueprint for how we're actually going to put this all together. And in tissue engineering, there are two main strategies, two different blueprints that scientists tend to follow. So on one side, you have the top-down approach. This is where you first build a scaffold, a sort of biodegradable frame in the exact shape of the tissue you want to make. Then you add your cells to it and let them grow and fill in the gaps. It's all based on the idea that if you get the overall shape right, the function will naturally follow.
On the other side, you've got the bottom-up approach. This one is more inspired by how we develop in the womb. You start by assembling the cells themselves, using different signals to encourage them to self-organize into the correct structure.
So let's take a closer look at that top-down method, because it's a really common way to do things. Step 1, you fabricate your scaffold, and it has to be biocompatible so the body won't reject it later. Step 2, you soak that scaffold in a nutrient broth that's full of growth factors to encourage the cells. Step 3 is the big one. You seed the scaffold with your cells. And finally, step 4, you put the whole thing back in that controlled workshop environment and just wait. You let the cells grow, divide, and form a brand new piece of living tissue.
Now, growing a really thin layer of cells is one thing, but building a thick, complex, three-dimensional piece of tissue, something like muscle or bone, that requires some more advanced gear. We need to find a way to supercharge the whole growth process. See, the problem is, if you just let a thick tissue construct sit in a dish, the cells in the middle can't get any nutrients. They're just too far from the liquid media. They'd starve. To solve this, engineers bring out the power tools of the trade, things called bioreactors.
So what is a bioreactor? Well, you can think of it as a really advanced, high-tech incubator. It's a special device that's designed to mimic the dynamic, active environment inside the human body. It can actively pump fluid through the tissue, it can stretch it can compress it, even send electrical signals through it, all to help create a much more realistic and functional final product.
And the benefits of using one are just massive. That fluid flow makes sure every single cell gets the food it needs and gets its waste carried away, which lets you grow much thicker tissues. And by applying those physical forces, like, say, stretching for a new piece of muscle or the feeling of blood flow for a new artery, you can actually train the cells to organize themselves and function more like they would inside the body. It's a total game changer.
So after all this work, the cells, the scaffolds, the bioreactors, what do we actually do with these engineered tissues? Well, it really boils down to two major applications. The first is implantation, creating things like engineered heart patches or new cartilage to go inside a patient and repair damage. The second is for what's called in vitro modeling. Here, we build small-scale living tissues, like artificial skin, so we can test new drugs or cosmetics without having to use animals or even human volunteers. And there you have it, the whole process, from the biological bricks all the way to a finished building. By carefully choosing our cells, recreating their supportive matrix, and following a very precise blueprint inside a high-tech workshop, tissue engineering is, quite literally, laying the foundation for a whole new future of medicine. Which brings us right back to that big picture. Now that you know the basic principles, you know about the cells, the scaffolds, the bioreactors, just think about it for a second. If you were the engineer in charge, how would you design a patch to repair a damaged heart? What kind of cells would you use? What materials would you build your scaffold out of? The answers to those very questions are exactly what scientists are working on in labs all over the world right now, building our future one cell at a time.